I'm Linda Tempelman. I'm currently the Chief Scientific Officer of Giener Life Sciences, and I want to tell you a little bit about our medical device technology. So we are working on an oxygenated cell capsule platform for cell therapies. So basically, if you have an allogeneic cell therapy you want to deliver to the body, and you don't want those cells to contact the body, you may choose to encapsulate them. We are developing an encapsulation system that will allow you to encapsulate the cells, oxygenate the cells because you've prevented them from having blood vessels touch them, and move on from there. So basically, I'm showing here some pancreatic islet, tissue, and whenever you're trying to put a lot of tissue into the body to treat something, this could be a good therapy. So just a little bit about our company. Our parent company is an expert in electrochemical technologies, and in the past year, we've spun off two companies, one to work in the energy sector, and then Giener Life Sciences, which I'll talk to you about today. So the parent company has been an R&D company for many years, a very large patent portfolio, working for many government agencies in a number of technologies. I used to work in the sensor technology area, and now I'm working for the past 15 years, more or less, in the oxygenated therapy space. So Giener Life Sciences is working in the diabetes space, so we have private funding to work in that space now, and one of the reasons I'm talking to you today is because we are looking for new partners with new cell treatments that need a way to deliver those treatments to the body. And our expertise is in packaging these in macroencapsulated cells and adding oxygen to the system. So Giener Life Sciences wants to partner with other companies that have unique cellular products, and they need a way to deliver them to the body. We also, if necessary, will in-license some cell technologies as needed, and we are working on some technologies where either the oxygen or the hydrogen that our system makes can be used as therapy itself. As I said, we're working in diabetes. We have some NIH grants, so NIH has been a great partner for us for early non-dilutive funding over the last 5 years in this technology area. Finally, we're planning proofs of concepts to move into other cell indications, and we will be doing a Series A raise in the 2020 timeframe to start working on other therapies besides just diabetes. So a little bit about our technology. So I think people are aware about macroencapsulation. So macroencapsulation technology is very good if you want to immunoprotect your cells and if you want to have a chance to retrieve those cells later, either because you want to reduce them or because of a safety profile, you want to be sure in the beginning that you can retrieve them if necessary. So in this particular capsule, we have an outer membrane that vascularizes with the body and an inner membrane, it's a little hard to do on the screen here, an inner membrane that protects the cells from the immune system. So blood vessels integrate to the outside of the capsule and provide nutrients and transport of your therapeutic drug out. It's only a minor surgical procedure to implant this subdermally, and the capsules can be replaced if necessary. Excuse me. So what we find is that there's been a lot of advancements in stem cell technologies, gene-modified cell lines, et cetera, and people have not been paying enough attention to how these will get delivered to the body. So one of the problems, however, with macroencapsulation, people have been working on macroencapsulation for 20 plus years. One of the problems we think is going on here is a lack of oxygen in the system. So basically, the rate-limiting nutrient in these capsules may be oxygen. And the reason being is oxygen is present in a very low micromolar quantity. Other nutrients can diffuse through the tissue, but we've had difficulties with oxygen not diffusing all the way through the tissue. So even when a cell has enough oxygen to live, you may find that the oxygen level is too low and the cells are kind of in a stasis state where they're not producing the product that you want. For example, if you don't give supplemental oxygen to the cells and you have the cells in a very dilute state, pancreatic islets, you'd require something about the size of your torso, 1 foot by 1 foot, to treat the person if you put the cells in the body very dilutely, and obviously that's not going to work. So in our case, when we have the oxygenated cell capsule, you can see here that in the center of a capsule that's about 500 microns in diameter. You see in the center of the capsule, we're supplying oxygen in the 500 millimolar mercury of a level, and then the cells consume the oxygen as a function of distance going out, and then out at the outside, everyone's excited the capsule is vascularized, but there may be only about an oxygen tension of about 50 out there. So this is the case when you oxygenate the capsule. If you don't oxygenate the capsule, unfortunately, you start at 50 at the outside of the capsule, and things just go south from there. And by the time you get to the middle of the capsule, you have a necrotic core, and that's the dead tissue that people have seen for many years in histological sections of macroencapsulation. So our enabler is to actually have an engineering solution of delivering oxygen to the center of the capsule. So what we show here is that we have an oxygenator that's about the size of 2 dimes right now. Our first design has been on bench testing for about 2 years now, generating oxygen. Of course, our company has made oxygenators for nuclear submarines that have been working for 10 years, so we're not really concerned about the catalytic component lasting for a long period of time. We've also had these in animal models for several weeks, and we've just started a long-term study of having these oxygen generators produce oxygen in the animals. Our second design is just getting fabricated now at our medical device manufacturer, and that pilot run will be completed at the end of the year. So basically, you take an oxygen generator and you have to combine it with a system. So the system would include the, thank you, I appreciate it. The oxygen generator is combined with cell capsules, and then the cell capsules are also combined with some electronic components. So to make oxygen from water, we harvest water from the body, and then we need power to do this. So the plan is that there will be a rechargeable battery, and the person would have to recharge that battery by strapping on a power pack for a few hours once a day. And the good news here is that you are able to use other cell types in our system besides pancreatic islets, which we've used so far, and you could use other cell capsules. We've been using an ePTFE-based capsule. And so why would you want to use an allogeneic platform? Of course, many people realize that if you have an allogeneic cell line, you can treat all people, and you only need one cell source. So that's an advantage over autologous systems. And we're looking at any application where someone doesn't have a therapeutic that can be oral. If you're already giving the person a therapeutic that's delivered by IV, our technology could be an example of that. And of course, patients that need multi-year treatment don't want to have percutaneous lines. So in 2015, we presented this material on a proof of concept in an animal study. So these are rats, and these are pancreatic islets that are human cadaveric islets in origin. And if you take the same number of cells and put them in a capsule, and you turn the oxygenator on in one case and don't turn it on in the other case, you'll see that you get a big difference in treatment of diabetes over a month. So basically, if you don't add oxygen, the rat maintains high blood sugar and they're not treated. And if you turn the oxygen on, in a large number of cases here, you see that we've treated the diabetes. And at the end of the 30 days, if you look at the capsules, only the capsules that had the oxygen on show pancreatic islets that are stained here positive for insulin. So this is what we've been working on recently. We have this technology protected by a portfolio of patents. One is issued in the US now, and then there's a family of 4 other patents that are already submitted. So some of these are for the oxygenation system itself, the electrolyzer components, and some are for the oxygenated capsule and then the whole system. Our company has a long history of experience with electrochemical technologies, and we're uniquely suited to do this work. So as I said, we would like to apply this technology as the platform technology gets brought up in our diabetes work. We'd like to be able to put other cell types in our system and tailor our system for other implant sites or other size capsules that would work for other things. And so here's some suggestions, some ideas, and as I said at this meeting and throughout the year, we're looking for people to partner with in this area. Here's our Giener Life Sciences team. So You'll see our team shown here. We've just hired a new CEO who has extensive medical device experience, including with Becton Dickinson for 14 years, and is a startup CEO recently. Andy Belt is an executive in all three of the Giener family companies, and I've been one of the co-founders of the company. And just recently this year, we've added to our to our board, and our board now includes outside members as well as corporate and owner members. And the way we do this work on a small team is we partner with other companies. And so some of the other companies we're partnering with is a company that has an ePTFE technology, Theracyte, and then Proven Process Medical Devices is working on our manufacturing with us. And we are using CBSET for our small animal studies, and we'll be using them for our large animal studies. So thank you very much for your attention, and I hope to take questions afterwards.